Hello, everyone. I'm sitting here with Dr. Dominic D'Agostino in Tampa, Florida. Dominic is an assistant professor at the University of South Florida, where his primary research focus is on metabolic therapies, particularly nutritional ketosis, ketone supplementation, and how they affect a wide variety of pathological conditions, ranging from neurological disorders like ALS to epilepsy to muscle wasting and cancer. So very, very interesting stuff that Dominic's doing here in Florida. He showed me some of his really cool equipment, which is super exciting and very interesting, some things I hadn't seen before. So welcome, Dominic.
Thanks for having me.
Yeah, thanks for inviting us to your lab.
Have you here in my lab, yeah.
Super cool.
Thanks.
So tell us a little bit about some of your more recent interests and research, where you're sort of been. What have you been looking at?
Okay. So more recently, I'd say within the last 3 to 5 years, and it took a while to develop them, but as you know, we're working on ketone supplementation. And the idea is to kind of mimic the therapeutic effects and performance enhancing effects of the ketogenic diet. So nutritional ketosis achieved through a dietary means with a low carbohydrate, high fat diet has therapeutic effects for a broad range of neurological disorders, in particular seizures. And you can make the argument that pretty much every neurological disorder is some way linked to a metabolic dysregulation. And we're interested in understanding how nutritional ketosis may help to preserve and sort of stabilize brain energy metabolism to metabolically manage these seizure disorders. So in the process of understanding and studying the ketogenic diet, we are developing a broad range of ketone supplements, which can include ketone esters and ketone mineral salts, where we combine a ketone body, beta-hydroxybutyrate, to an essential electrolyte and create a wide variety of salts. And a lot of the work now is focused on formulating these to make them tolerable, to make them palatable, and to understand their therapeutic potency for different types of disorders.
Yeah, so you mentioned that at the root or at the heart of a lot of these neurological disorders is a metabolic dysfunction, and how, first of all, when we talk about nutritional ketosis, to me, that's such a broad way of describing.
Very broad, yeah.
Like, what is nutritional ketosis? And how do you achieve it?
Mm-hmm. So nutritional ketosis could be, it's defined, it kind of has the same definition in my mind. It's achieving and sustaining a level of blood ketones. And I think when people say they've done the ketogenic diet, they did that, it didn't work for them or it did work for them, I would ask the question, well, did you measure blood ketones? Did you confirm, in fact, that you were able to achieve a state of ketosis, defined as an elevation of blood ketones above 0.5 millimolar at the very least? Ideally, you want to stay between 1 and 3 millimolar of ketones. And when you've achieved that state,
you're talking about blood ketones?
I'm talking about blood ketones, yeah, which is kind of the gold standard. And this can be measured. There's a number of different devices out there that measure blood ketones. And when the state of nutritional ketosis is achieved, you're also not only that biomarker is kind of there and we have technologies to measure it, but it would also be important to measure your blood glucose and maybe insulin levels too. So when the suppression of the hormone insulin drives hepatic ketogenesis. It drives the body's ability to make ketones. And that has therapeutic implications for type 2 diabetes, obviously. So my definition of nutritional ketosis would be an elevation of blood ketones, and the difficulty in prescribing that or telling someone to do it is that the way to implement that is kind of similar with everyone, but everyone responds differently depending on where you're coming from. So if you have an obese subject that's type 2 diabetic, it's going to be different than an athlete, or different. And women, there's some differences between women and men, I think. So defining nutritional ketosis is relatively easy with an elevation of blood ketones, but implementing it and being able to, for the individual to commit to it and have that kind of ability to control their diet, which is very linked to lifestyle, has been sort of a difficult thing to do. And that's where ketone supplementation kind of comes in and can allow someone to rapidly achieve nutritional ketosis and sustain it, and perhaps get many of the therapeutic benefits that we're just finding out now. We know that ketones are more than just the metabolite. They're more than just an energy metabolite that the brain can use, but they are metabolites, especially beta-hydroxybutyrate, are very powerful signaling molecules.
Right.
And we're just beginning to understand sort of the therapeutic effects of these metabolites as signaling molecules. And that's a big thrust of our lab right now.
Oh, you're interested in looking at this?
Yes.
Interesting.
Yes.
Yeah, I'm familiar with some of Eric Berdine's work at Gladstone and how he's the science publication.
Pioneer in
it's very.
area.
I'm very interested in beta-hydroxybutyrate as, not only as a source of energy for mitochondria, so being able to be converted into a thermodynamically favorable source of energy, but also the fact that it's able to change cell signaling in the brain, and it's able to turn on genes that are involved in dealing with stress better. Some of these genes are involved in longevity, FOXO3 for one.
Absolutely.
So it's all very interesting. But something that I like in my mind that I'm not exactly certain about is that nutritional ketosis. So, eating a high-fat diet. And of course there's, okay, well, what types of fat are you eating? Are you eating more polyunsaturated or eating more saturated? How much protein are you eating? What types of carbohydrates? Are you getting fiber? I mean, there's so many different diets, very complicated. But there is no doubt that there are interesting therapeutic effects from nutritional ketosis. I am interested in the actual end product, which is ketone bodies and these, signaling molecules like beta-hydroxybutyrate. Being able to get those maybe even without having to eat a high-fat diet. You can get them from fasting, from intense exercise, right? Also, you can achieve
post-exercise ketosis.
Post-exercise. Exercise.
Absolutely.
So I guess kind of the question I had for you with nutritional ketosis, but besides how do you define it, was what do people eat to obtain that? Like, how do you get blood ketone levels between 1 and 3 millimolar? Like, what do you have to eat? What do you not have to eat?
Okay, that's a good question, and it varies depending on who you talk to and what's optimal. I think a good way to approach it would be to describe what has been used classically and how the diet has evolved over the last 2 to 3 decades. I think with some of the work with Eric Kosoff at Johns Hopkins has kind of advanced the idea of using a modified Atkins diet or modified ketogenic diet. And I can kind of talk a little bit about strategies that I use, I think, and others that I know use to achieve that state and sustain it for optimal performance and health, I think. So in taking a step back, the classical ketogenic diet would be like a 4-to-1 or 3-to-1 diet, and that's kind of the ratios of the macronutrients, 4 being fat and 1 being a combination of carbohydrates and protein together. So pretty protein restricted diet. And the ketogenic diet is not a high protein diet. It's actually moderate to low protein diet. And most people don't understand that. They think, especially in the fitness community, if they go on a ketogenic diet, if they say they've been on a ketogenic diet, what they will describe to me would be a very low carbohydrate, high protein, moderate fat diet. But the ketogenic diet, as it is used classically for drug-resistant epilepsy, the original, was like 90% fat, like 85 to 90% fat, and maybe about 10% protein, typically, and 8 to 10% protein, and very minimal amount of carbohydrates. And it was heavily based upon the use of dairy fat. So dairy was the primary kind of vehicle, to get calories into you. And we know that the use of dairy, especially in some people, dairy protein in particular, but even sometimes dairy fat too, can have negative consequences for some people. So there's a wide variety of kind of ketogenic diets out there, And the ketogenic diet has been defined by this ratio of macronutrients. But what we're learning now is that it's more than just macronutrients to optimize the diet, especially for individuals. The sources of the macronutrients, the fatty acid profile, the type of protein, it allows for some amount of carbohydrates, and the types and quality of the carbohydrates as it relates to gut health and gut microbiome, I think is really important. I think it's really important to optimize the diversity of carbohydrate sources in the form of raw vegetables, and I think that can optimize the diversity of the gut microbiome too. I think those two are linked: the diversity of the foods in your diet and the diversity of your gut microbiome. I've seen that just through feedback. It's not really studied, but it's something everyone knows and knows should be studied. So the classical ketogenic diet is very strict to follow. There's a few studies showing that it can influence a lipid profile in a negative way, meaning high elevation of LDL. And in kids, I think that followed the diet, they had a high level of triglyceride. There was one study that's often referenced in regard to the ketogenic diet being atherogenic. The triglycerides were really high in some of the kids.
There's probably a very complex gene-nutrient interaction as well. There's some, I'm not sure if you're familiar with any of this work. It's something I'm getting into recently, is this nutrigenomic field, and particularly there's PPR gamma and PPR alpha, which is important for ketosis. There are polymorphisms that are associated with not being able to respond well. So it's very likely that some of these people that don't respond well or may have that polymorphism.
Yeah, undoubtedly, because there's some people that really have a negative effect. But I would say it's kind of few and far between, maybe as high as 20% in some circles. But generally speaking, if you have a dietitian that's pretty savvy and has worked with enough people, I really emphasize experience, because there's no substitute for experience. You could know as much, you could know all the biochemistry behind nutritional ketosis, all the pathways or whatever, but working with a dietitian, I always stress that in people that want to try the ketogenic diet, to start off with a good dietitian. They can tailor and tweak the diet for your specific needs. And I find that for me personally, a diet that's low in dairy is really essential, because I just don't do good with dairy. And nuts have been a staple for some people for the ketogenic diet, and I have like a mild nut allergy, so that's another food that I've kind of had to eliminate. But despite that, I've been following what I call a modified ketogenic diet, and I think it's pretty similar to the modified Atkins diet that Eric Kossoff has been using at Johns Hopkins. He's at Johns Hopkins, mainly a neurologist, but has a team of dietitians working with him, and has done an incredible job advancing sort of the use of the ketogenic diet. He follows in the steps of John Freeman, who was really a pioneer in getting the neurology world to recognize the ketogenic diet as a viable metabolic therapy for drug-resistant epilepsy, and did a lot of work with the Charlie Foundation and work with them to help promote awareness of the diet, education of the diet. So some work that Eric Kossoff has been doing over the years and publishing on is showing that the modified Atkins diet has much of the therapeutic potency of the strict classical 4-to-1 ketogenic diet. So instead of 90% fat, the modified Atkins diet is roughly 65 to maybe 70% fat, which is kind of what I follow now, and about 20 to 30% protein, with the balance being still very low in carbohydrates, like no more than 5 or 10% carbohydrates.
Okay.
But it's more liberal in the use of protein. And it's also advocates the use of medium chain fatty acids can be incorporated into the diet. So by following a diet that's more liberal with the protein and less not restrictive, or less in less of a need of such a high fat content, I've been able to maintain a moderate state of ketosis, as do many of the therapies, or many of the patients that are benefiting from this therapy for a wide range of disorders. So I think that makes the diet more accessible to people. And I think the biggest hurdle now is just compliance. So people know the benefits of the diet. Some of them are a little concerned with the side effects of the diet, as far as lipid profiles being altered. But
what about gut?
the gut, yeah.
Have you been looking? So how long have you been doing this modified ketogenic diet?
It sort of evolved from 2008 to 9. I started experimenting with it because that's when I got interested in using.
And you've continued on ever since then?
Yeah, I've never really
quite a long
gotten out
time.
of ketosis. Yeah,
That's quite a long time.
If I did, I may have reverted back to like a low carb paleo, which is, maybe up to 40 or 50% fat or protein on some days.
Yeah.
So I've been a little more liberal on the protein at times, but yeah, I've been following it for a long time, and I do blood work.
Yeah, the
really
reason
consistent.
I ask is because it's the one, like I mentioned, I'm very interested in nutritional ketosis, which is why I'm excited to talk to you, because you know so much more about it. And I'm trying to get to the bottom. There's this conflict in my head with eating a lot of fat and its effects on the gut, because I know a lot of gut researchers. A lot of my friends are looking at gut health, and one of the main things that you do to induce endotoxin, which is released from ectotoxin membranes, one of the main things you do to induce gut permeability is you give a high-fat diet. You feed mice. This is all done in animals, of course, which is there's lots of problems with that.
And what's the typical fat source for that? Is it lard, or is it?
A variety of different fat sources.
Yeah.
I mean, there's lard, there's corn oil. I mean, so there's a variety of different fat sources. But fat itself, in order to be digested, you have to make these bile acids like deoxycholic acid, which causes endotoxin release. It also acts as a surfactant. I mean, it's like a detergent. So I'm not convinced that it's not not healthy, but I'm sort of trying to get to there's a disconnect in the literature, because there's so much information out there showing the benefits of a ketogenic diet, nutritional ketosis.
Let me ask this question real quick. So when endotoxin is released, from these bile acids, so there would be a characteristic cytokine profile that would reflect that, right?
Yes.
And okay.
So there's a cytokine profile, and there's also, you measure endotoxin in the blood, which is a very tricky thing to do because there's a lot of false positives.
Yes.
And I know someone who's trying to develop an assay to make it actually, because he's very OCD about it, and it's like it's not out there for clinical use yet because of that reason. And even doing it in animal studies, there's a lot of researchers that don't really do it right, measuring endotoxin.
Is there any benefit to endotoxin? So when I go exercise, right, and you measure my blood, and we look at reactive oxygen species or inflammatory mediators, you could look at the blood and say, don't do that. This is not a good thing to do. Whereas, if you have periodic spikes, maybe an endotoxin, is it stimulating a hormetic effect where it's enhancing my resilience or resistance to toxins?
Right.
Do we know that?
So endotoxin, I would say, based on everything that I've known and researched, and from my interactions with people that have been, doing this research, it's not like reactive oxygen species where there's, it's a potent signaling molecule that has this hormetic effect. It increases mitochondrial biogenesis. It increases all these genes involved in dealing with stress. There's lots of benefits. Endotoxin released from the gut, one, it causes more VLDL production because VLDL soaks it up. Yeah. So that's part of the reason why inflammation, is also correlated with an increase in LDL number. Yeah. It also binds to ApoB. It binds to the part. It binds to LDL, where the LDL receptors bind. So that LDL can't be recycled as well. So it kind of prevents. There's a lot of bad things about endotoxin being released. Now I don't know. Maybe there is some sort of slight benefit from endotoxin being released. I don't want to get too into the endotoxin world. I'm just saying that there is I'm trying to understand in my mind how
So you might be a little bit biased because the high-fat diet work has endotoxin. I need to get educated. I mean, I understand endotoxin from kind of like a basic science point of view.
Sepsis bad.
yeah, from sepsis bad. But I do know, when your body's challenged, even things like radiation. I mean, there's some things, I don't know of any case where an autoantibody is a good thing, right? But I know most of the things that are bad out there do have some benefits. It's the level.
Absolutely.
It's the level.
Dose is very important.
And the phenotype's ability to adapt to that chemical, that stimulus is really important. Like old people don't adapt to, older phenotypes don't adapt to, an oxidative stress stimulus as robust. So I would be interested. And there's ways to simulate endotoxin, right? Experimental models, and that's something that we could do. Challenge, perhaps, run an experiment where we have animals on different diets, where we can challenge them and look at inflammatory cytokines. We're doing a lot of that work now.
And gut health in general, gut permeability. I would love to see
we're
some
doing that too.
Research go in that direction because there's so many unanswered questions in my mind. And there's a lot of bad data out there, from the high-fat diet and the effects on the gut, because when you look at control diets and the high-fat diets, the control diets have 50% more fiber, and it's that's not really the same thing if you're just changing, fat. So you have to sift through all sorts of crap, and it's there's lots of data that you just have to throw out, but still at the end of the day, I'm uncertain, and I'm weary about me eating a high-fat diet. Not to mention the fact that I have certain gene polymorphisms that may not be as compatible, but because of that, because of gut health, that's my major limiting factor for me is gut health, and I would like to see more research on ketogenic diets and gut health. That's something that would be, I think, is important.
How to optimize it.
Or just what the effects are. Maybe for one, you're starving out probably a lot of the pathogenic sugar-eating bacteria. So there's probably some good things going on. And then there's, what effect does medium-chain triglycerides have? Is it the same as long-chain fats? Maybe not, right? So there's, different types of fat. There's studies that have been coming out very recently showing the effects of polyunsaturated fat on gut biome and how it has a positive effect. So maybe if you eat, certain types of fat, more of these types of fat versus, there's lots of things out there to be explored.
Shifts it. Shifts it. I mean, your gut microbiome, has an appetite for whatever you're feeding it, and that probably influences profoundly your own appetite. So I think if you have sugar-eating gut microbes, you're probably going to be craving sugar if you go, a few hours without having sugar.
Yes,
because and I think I would be very interested in not only shifting someone from a high carb diet to 2 or 3 different types of ketogenic diets. I think would be important with specific fatty acid compositions and fiber compositions. And I think it's the diversity of the fiber that's incorporated.
Diversity, yes. It is very important. There have been, there's lots of different types of bacteria, and they're eating different precursors to generate a lot of these other signaling molecules, short-chain fatty acids and things like that are regulating your immune system. They're regulating, literally hematopoiesis, Tregs, natural killer cells, lots of, and this is a whole blooming field of research. That's also another reason I've been a little hesitant to experiment with this, because fiber is so important to me. Fiber from vegetables, I'm not adverse to fiber from fruits. So fiber from fruits, legumes, beans, I like to get a lot of fiber in my diet. And so I don't know if that is compatible with it.
I get more fiber on a ketogenic diet, we're talking about a well-formulated ketogenic diet, as Jeff Volek likes to, and because that is really important, because you talk to people that eat ketogenic diets and it's all over the board. But I think a well-formulated ketogenic diet would have an abundance of fiber sources from everything from green vegetables, of course, but would include a salad. And I think it's really important from a gut microbiome perspective to get in raw vegetables. I think from what I've known and the half dozen individuals that I talk to that are kind of experts in this field, they think that has a pretty profound effect. And I've always done that. And I would say my gut health has not, it maybe due to what I eliminated in my diet. I grew up in an Italian family eating a lot of pasta breads. They were the staple foods. And I gravitated toward a paleo diet, mid-90s, early 2000s, and then the ketogenic diet. And I have never had better gut health than when on a ketogenic diet. But my diet is, like I said, more of a modified Atkins and has a pretty liberal amount of vegetables in it. And I think the benefit to including the vegetables, they're just carriers for the fat. And they also slow protein digestion, which helps minimize the insulin spike that you can get from protein and helps keep me in ketosis. But I mean, getting back to your question about the ketogenic diet or a high-fat diet influencing endotoxin or factors, I would think that would show up like in the literature that if some, and maybe it does, but usually a high-fat diet is in the context of a high-carbohydrate diet too. So that's what we don't know. And when we talk about, LDL particles being elevated, like even skyrocketed, LDL little p, that is only understood in the context of a high-fat diet, which also includes sugar, like a Western diet.
Absolutely.
So we don't yet understand lipidomics and the shifts in lipid profile of the ketogenic diet. LDLP specifically, we don't understand it in the context of keto-adaptation. So
okay, what's that?
Keto-adaptation, it's kind of a term that Jeff Volek and Stephen Finney coined, but I think it's very descriptive in my mind as a physiological process when your body has adapted to using fatty acids and ketones for fuel, where you've biochemically, physiologically shifted your metabolism from burning glucose as the primary fuel to burning glucose and also equal or maybe more in some cases ketone bodies, primarily from your central nervous system. So when that happens, acutely you get an elevation of blood ketones, and over time what you do is get an upregulation of the transport mechanisms, so your ability to sort of make ketones, utilize ketones, and metabolize ketones in the cell is dramatically increased, as is the oxidative capacity of your cells.
After you start making more ketones?
It's sort of driven by being in a state of ketosis, enhances fat oxidation over time. So when we say keto adaptation, we should probably say keto and fat adaptation. Okay. So there's studies out there that show our metabolic physiology changes pretty profoundly from eating this. We burn what we eat, right? So a high carbohydrate diet, we are pretty much burning that as fuel, and our bodies can adapt remarkably well to burning a macronutrient profile that's reflective of the ketogenic diet. And when we do that, a lot of really remarkable beneficial metabolic processes happen, including mitochondrial biogenesis, reduction of ROS, reduction of inflammation, a reduction of specific inflammatory cytokines that may be associated with age-related chronic diseases are reduced.
Interesting. So reduction of ROS, because I guess some of Richard Veitch's have put some ideas out there about the
ketones direct effect.
Because it's so funny, because in my mind I always think about if you're inducing your mitochondria to work more, you're going to make more reactive oxygen species, right? And that's sort of what I think would be a driver of killing abnormal cells, cancer cells that are primed to die. They're expressing way more proapoptotic proteins.
Yeah.
But they've countered it with anti-apoptotics. All they need is a little push, a little reactive oxygen species push to death.
And that's increased initially.
It is, okay.
So it actually activates NRF2 and stuff. So when someone gets on a ketogenic diet, it's a stress to the body.
Yeah, so you are making more, because if you're causing your mitochondria to only work, right, that's the only way you can make energy, then you think that you have to be making reactive oxygen species.
Yep.
At least if you're looking in the context of a cancer cell, which would be like lytic and not using their mitochondria at all, right?
Yeah.
Then definitely there would be a much more of an increase in ROS. So ketones. So you're saying that it can suppress ROS. Is that through some of what Richard Veitch has put out there in the semi-ubiquinone, how it's like
oxidized Q makes it less available.
Because that's the most.
If Q is oxidized, So
Q being ubiquinone.
Yeah, ubiquinone. If ubiquinone is oxidized, which is achieved with our beta hydroxybutyrate metabolism, if that's oxidized, then you have less availability for that electron to react with molecular oxygen in the metabolic pathway. So you would produce less superoxide anion, which is your precursor to more reactive oxygen species. And that's been shown elegantly in a number of models, including the cardiac model, which he did, the Langenhorn model, the perfused heart preparation, showing that you get a greater hydraulic efficiency of the heart in the presence of ketones. With a given amount of oxygen, you can generate proportionally more ATP, energy currency.
Yeah. Is there another way that ketones also suppress ROS? Or is that through the
acutely
hormetic effect of activating mitochondria, then increasing NRF2 and things like that?
Yeah, that would be a secondary effect. I would say from an acute point of view, as simply as a metabolic fuel, the mitochondrial efficiency is greater. So you will have a greater mitochondrial membrane potential, a greater driving force for ATP synthase to make ATP. So it energizes the mitochondria in a way that would be expected from a metabolic fuel that's sort of superior from a bioenergetic point of view. So you have a greater capacity to generate ATP for a given amount of oxygen that's available. So with that occurring, the metabolic efficiency of the cell would be sort of preserved. You're using less oxygen to make the same amount of ATP, less reactive oxygen species. And of course, if you're shifting away from glycolysis and shifting more towards oxidative phosphorylation.
So in the context of a cancer cell.
In the context of, any kind of cell, like your tissue, really, skeletal muscle or cancer cell. Yeah, you are forcing the body in a way, and it's a stress initially, to upregulate mitochondrial machinery, really. And more mitochondria will start, budding off and creating mitochondrial biogenesis.
Mitochondrial biogenesis.
Yeah, the number of mitochondria, the proteins that are associated with the electron transport chain, those proteins are upregulated. So you make more of these proteins.
Yeah, it acts very similar to lactate. I don't know if you're familiar with any of the brain work and George Brooks lactate.
Very interested in lactate as a graduate student. Yeah, I'm
goes
studying
to the same transporter, right?
Yeah, MCT. I studied lactate as sort of an undergrad and graduate student, like I am studying ketones now as an alternative fuel.
Were you looking at it in the brain?
Yeah, during hypoxia. So I studied the neural control of autonomic regulation, so brain hypoxia and what our brains do under hypoxia. And lactate is a big player in preserving, brain function, viability, health, and I studied lactate as an alternative energy source under hypoxia. And now I think of ketones as like the alternative energy source, when your brain is under normal physiology. And I think there's some uses for lactate too as a fuel. I think when we exercise, we're creating lactate and we feel good. It's not really talked about. It's something that I want to study and maybe talk about a little bit more, is that lactate, you're also sending, not only are you sending ketones to your brain, but you're sending lactate to your brain. And I think that's maybe not talked about that much, but there's potential out there for lactate supplements.
Yeah, George Brooks at UC Berkeley, I don't know if you're familiar with any of his work.
A little bit.
He pioneered the whole lactate shuttle theory, but he's been looking at the effects of, lactate generated during exercise, for example, when you're forcing your muscle cells to work harder and you're making more lactate, it crosses over the blood-brain barrier, gets into neurons. So neurons themselves actually use lactate generated from astrocytes. So they're using neurons are actually using lactate. It's also a thermodynamically and energetically favorable source of energy, much like ketones. And so neurons like doing that because, one, it's easier, and two, because glucose can then be freed up to be, shunted in the pentose phosphate pathway, which can be used to generate NADPH, which is, important for glutathione recycling,
capacity,
which makes sense. Well, I think there's a lot of parallels between, ketone bodies and ketone supplements and how they're not only being used as a preferential source of energy in the brain. Do you know anything about this? About like how it frees up glucose then to be used for other metabolic
Glucose-sparing.
pathways. Yeah, glucose
Yeah,
sparing.
yeah. It's thought that some of the work by Stephen Cunnane, I think, is shedding light on this too. If you
Where's he at?
he's in Canada.
Canada, okay.
I forget the institute that he's at. Joe Lamanna has done some similar work, and he's at Case Western, and looked at the kind of the interaction of what does the brain prefer as what's the preferred fuel source for the brain. I get this question a lot. I think it depends on, you know, I don't know if it's right to say that the brain always prefers
The brain
ketones.
cell we're talking about.
Yeah,
for one.
the brain, yeah,
Neuron or
and
astrocyte.
neurons and astrocytes. But yeah, I guess, I mean, maybe we're definitely biased towards understanding neurons relative to astrocytes.
I think most
So
people that ask you that question.
astrocytes are fascinating.
talking
I think
about neurons.
we need to study that more. But I think in the context of like aging, in the context of traumatic brain injury or pathology, I think the brain will really prefer to use ketones because, or in the context of some kind of stress, you know, hypoglycemia or something like that, I think the brain will also prefer to use ketones.
Yeah. So why do you think that is?
Well, I think,
I have my own
yeah,
reasons why.
there's a whole host of reasons that, or things that can cause impaired brain energy, brain glucose metabolism. And that could be internalization of the GLUT3 transporter, which occurs, it's kind of linked to Alzheimer's pathology. GLUT3, yeah. And there's a couple key enzymes that are either deficient or not active like they should be. Pyruvate dehydrogenase complex is deficient in. Like,
so wait, are these are the GLUT3 changes in astrocytes or in neurons?
Primarily in neurons.
Neurons.
Yeah.
Okay.
So the glucose transporter in neurons is not.
But so if neurons are using lactate primarily, the astrocytes are actually what's
shuttling the lactate.
Shuttling. The astrocytes are using glucose, and that's why the brain needs glucose.
Yep.
And they're producing the lactate.
Yep.
The neurons are using the lactate because it's getting shunted and converted into pyruvate.
And the contribution of that is not completely known, but it's pretty significant contribution during hypoxia, I would think.
There's been studies out there showing that not just during hypoxia, but actually just under normal physiology, that
it's a key player.
It is, yeah. The neurons are getting lactate continually from astrocytes, which are generating it, but that the astrocytes become aberrant during, for whatever reason, it's not known. Alzheimer's disease, traumatic brain injury, so they can't make that lactate, and neurons have to start using the glucose, which then is, but I mean, that's just one.
Yeah, I've heard about that. I haven't looked into that. Yeah, so there's some key enzymes in that people look at, not only just the lactate levels.
I think that, I have no idea what they're looking at, but for the pyruvate dehydrogenase complex, you're saying that's also aberrant in some of these disease states, because then you wouldn't be able to use the lactate because you couldn't convert it into pyruvate. So anyways, it's all kind of, I totally interrupted you though, but you were talking about, what were you talking about? The aberrant enzymes like GLUT3 and pyruvate dehydrogenase, and how these are aberrant in different neurological disorders.
Yeah.
Okay, sorry.
I think, well, maybe the question that we're trying to get at is the contribution of ketones as a brain energy source, and especially in sort of in academia and circles where I teach, it's still not really known as it's not really accepted or understood that much. But I think the capacity, the metabolic flexibility of the brain to be able to use ketones, we can exploit that, and we do that with nutritional ketosis, and it's altering brain energy metabolism and the neuropharmacology of the brain in ways we don't completely understand now, meaning the neurotransmitters in the brain. So I could draw off GABA. So you have more GABA. The GABA to glutamate ratio is shifted in favor of higher GABA. So there's a higher GABA to glutamate ratio when one is on the ketogenic diet. And
in people that are not, having normal people as well, just like in general?
Yeah, it's thought that, and there's different reasons for that. There's glutamic acid decarboxylase is an enzyme.
Is that how it affects? Is that how it helps with epilepsy?
That's part of it. I think there's about 20 or more things going on as
Right.
really, if you look at all the signaling pathways,
As does everything
actually
in
what happens, with metabolism. But I think a key player in that is an elevation of GABA to glutamate, and we need glutamate to make GABA, right? But the enzyme is elevated, and the pathways are shift in favor of more glutamate to GABA, which has a stabilizing effect on your cell membrane and neuronal activity in general.
How readily do ketones cross the blood-brain barrier? Is it?
It's thought, up to 5 millimolar, maybe 6-7 millimolar, they can readily cross the blood-brain barrier.
You have to get that high before they start crossing?
No, you start impeding. Once you get levels up that high. And it hasn't really been studied, in depth. We did brain metabolomics to show that, when we looked at the ketone levels in brain tissue and ketone levels in the blood, and there's a high correlation there. Interestingly, we had a diet too that was high in medium chain fatty acids, and although I heard medium chain fatty acids could readily cross the blood-brain barrier, long chain fatty acids typically don't. Short chain fatty acids sort of do, but we found very high levels of medium chain fatty acids, indicative of them. And these were normal, healthy animals. So when you take medium chain fatty acids, they are readily, from the looks of our metabolomic data, just readily crossing the blood-brain barrier and capable of being used as fuel.
It's fascinating.
So, which is very, it was really interesting. I heard about that, but when you see the data, at
so when
really
they're
high
in
levels.
the brain, then how are they? So then what happens? So they're used.
Their medium chain fatty acids can be oxidized, just like fatty acids for fuel. Through oxidative. But ketones are also very high in the brain, and to really capture and understand it, you have to sort of capture the brain tissue really quick and freeze it, and then do an analysis, because the brain's using it as fuel after you take it. Your brain's kind of still alive. Sort of the stuff that I was showing you in the lab, where we can take the brain out and cut it like a piece of bread, and then record from it, so that brain is still active. So it sort of uses the metabolites. So to get a very clear snapshot of the difference in tissue versus blood is a little tricky, as is getting ATP levels in the brain. It's a little tricky. We're working on that to be able to do that. Another question, which I think will involve radio tracers, is actually to do like a ketone PET scan, where we can look specifically at the regional distribution of ketones and figuring out what areas of the brain may be preferentially shuttling them and using them as fuel, and how that may change under different pathologies, how that may change under oxygen deprivation, glucose deprivation, high oxygen, as I study oxygen toxicity.
Please look at genetic states like ApoE4.
Yes,
because that's one thing.
That's a biggie.
I'm very interested in some of this work you're talking about in ketone supplements helping with neurological disorders like Alzheimer's. It's been shown that is dependent on ApoE status, and that just it's disappointing to me, because I really want to understand why. And I just do you have any idea?
You're referring to the Henderson paper where they looked at Axona, AC1202. So the finding was in that study, which is relatively small, that at least not with the diet, but using a ketone supplement that was formulated with 20 grams of medium chain triglycerides. So they gave it to their patients, I think just once a day. And they did show fairly convincingly that the elevation of beta hydroxybutyrate correlated with an improvement in cognitive function. But that correlation was not observed in the ApoE4 positive group, which was a little bit, it's kind of surprising. I would like to see that study done using the ketogenic diet, or maybe the ketogenic diet formulated with ketone supplementation. And but so the question is, it didn't have a negative effect, but the question is, why didn't it have a positive effect?
Was there not another study where they actually did the ketone supplement, beta-hydroxybutyrate? I thought there was, but I could be mistaken just from my lack of understanding. Because that
C8
would shed
is
some more light.
ketogenic. Yeah, they use caprylic triglyceride, C8, which kind of makes a lot of ketones, relatively speaking.
Unless the ApoE is changing the way you're making ketones, right?
Good fatty acid.
But they measured. You said they measured beta-hydroxybutyrate in everyone, and
the
levels were the same. Levels were the same because it's like I want to understand why.
Yeah.
Why there was no positive benefit in ApoE4 carriers, because that's the one thing that it seems so promising, right? That these potentially ketone supplements or a ketogenic diet could help modulate, Alzheimer's disease.
Yeah, and I think you could do it in different ways. My student presented yesterday. She presented this week, but she graduated with her Ph.D. yesterday, and her work showed that there's a remarkable increase in blood flow. And previous work had shown that ketone bodies can increase brain blood flow by 30 to 40%. So that's another, when you have vascular dementia, when you have a decrease, that staying in a state of
or just brain aging in general.
Yeah, can have a profound effect.
Did she understand any of the mechanisms, or do you know? Can you talk about it?
Yeah, we're looking at that. Well, we did ischemic wounds, which, I guess you could kind of relate to an aged brain, right, with clogged arteries. We did a Doppler blood flow measurement and showed that it spiked up considerably when we can elevate ketones. One of the things, it was not dependent upon VEGF. So we looked at all the different factors. So VEGF was not increased in the wound. And we looked at a couple other things that we thought would be increased. The one thing that stood out in the data was adenosine. So adenosine levels are significantly elevated. Now, adenosine is sort of thought to make us sleepy. When we drink coffee, it's like adenosine receptor antagonist. There's high levels of ATP sort of being used, and we think that they may be causing an elevation of adenosine. But we really have to delve into the metabolomics to understand why that's happening. Regardless, adenosine is a very powerful vasodilator, and it's in higher concentration significantly in the blood, and that may be increasing the perfusion of tissues, peripheral tissues. And I think did
it have any effect on the blood-brain barrier at all? Did you look at that?
We haven't looked at that. My colleague has been kind of looking at that a little bit. We know with fasting and the ketogenic diet that you can increase permeability to the blood-brain barrier. Things get through faster.
Ketosis through fasting, or
both?
Combining the two?
Sort of, fasting-induced ketosis, or even the ketogenic diet can help increase the permeability of the blood-brain barrier to a wide variety of things. So if you are, sort of the implications from his perspective, that if you're getting a chemotherapeutic drug, if you're getting some kind of drug that needs to cross the blood-brain barrier that's impaired in some way, you might be able to get that across faster in a state of, fasting ketosis or the ketogenic diet. So maybe if you're fasting, maybe, there's just less stuff in your blood, there's competition of things getting through, and your blood's sort of clear, and you introduce the drug and you get more rapid transport because there's co-transporters and other things, or things that might be in your diet that may be impeding transport. There's multiple independent lines of evidence to suggest that being in a state of ketosis can help better transport things across. But when we talk about permeabilizing the blood-brain barrier, that's like a bad thing, right? Making it more permeable. But in general, though, you get a knockdown of neuroinflammation from the diet and from therapeutic ketosis, which is something the epilepsy world is very interested in. So there's this PET scan technique that allows us to look at neuroinflammation in the brain.
Yeah.
And we know that this is a conference that I recently came from, that may be an excellent predictor of when someone's going to have a seizure.
What would be really cool to do is possibly see if some of those effects are mediated through this pentose phosphate shunt, because glutathione in the brain is one of the major ways of reducing inflammation, right?
Yeah.
So let's say you do this in an animal model and then you inhibit some of the pentose phosphate enzymes that are, using glucose now instead to make NADPH and see, is that mitigated? Do you not have that same effect? It'd be interesting to see if that's part of the mechanism, because that's
it undoubtedly
really,
is. Yeah,
probably, right?
It's part of that, and so it plays into all that.
So I have another question for you. Thanks for letting me jump all over the place because I'm super interested in this and don't.
Yeah, we need to follow up on the studies. So one of the studies is sort of we want to do, just following back up on that question, is where ketones are being metabolized and what their contribution is to normal brain energy metabolism and disease states and hypoxia states and traumatic brain injury. So it's a lot to do. But
and there's overlap too.
That's the direction of where we're going with our research, is understanding that.
Just to jump back again before I ask you this other question to the George Brooks thing, is that he's doing some interesting work working with some physicians at UCLA, I believe, with traumatic brain injury. So these are people that come in with head gunshot wounds. So and they are looking at infusing lactate because so he's the lactate guy. Yeah. But ketone bodies would work the same way.
I think it would work better,
maybe even better.
Yeah, right. I think a cocktail of the two would be, and I've been debating whether or not to really study lactate sort of the same way I'm studying ketones, and maybe make lactate esters or lactate, and just to see what kind of results we see, starting with our oxygen toxicity model, and then working from there, which is an excellent model.
Obviously, with the lactate, you have to have intact blood-brain barrier. No oxygen's getting there, you will get then lactic acid buildup, right? Yeah. Because then the mitochondria aren't going to be able to use the lactate, which is converted into pyruvate as energy, so it builds up. But he has been doing some work and looking at this glucose sparing in the brain, and so of course ketone bodies would work very similarly. In theory. I don't
Do you know how he gives? Does he use a lactate? There's alpha L polylactate, I think. That's in a sports supplement and some other things.
He actually designed. I think he's the one that he's got a patent on that.
I was using that back in 1991 and
Cytomax,
2.
Right? That's his. Yeah.
When I raced mountain bikes, that was my go-to supplement, alpha L polylactate.
You should get in touch with George. He's great.
I know. Wow.
Yeah. So he. Let's see. What was I just going to say? I lost my train of thought.
The delivery or the.
Oh yeah. So the delivery. That he's doing in this specific study. They're doing it intravenously. So. Okay. But. I was mentioning, the blood-brain barrier has to be intact, otherwise it can go the opposite way, where it's actually bad for you. But it'd be really interesting to see, so the question is, one, will does lactate or, beta-hydroxybutyrate or whatever, ketone get into the MCT shuttle better? So there's questions of, which one gets in there, and can they be used together, and just all sorts of interesting things. But okay, so that was I'm sort of interested in this for my own personal.
The neuroprotective capacity seems really, compelling. I mean, from what we know about lactate, I'm approaching it going back to my old Ph.D. studies or whatever during hypoxia, but it would relate to so many traumatic brain injury as hypoxia. So you're mitigating the damage.
And also traumatic brain injury is inducing damage, and so glutathione is one of the major ways of mitigating that. And there's been studies showing that early on it's important. If you get that early on, it prevents the whole cascade. So I think that ketones and lactate both seem to have huge potential for glucose sparing, but that
I think chilling the body and infusing ketones, lactate with a couple few cofactors that are part of the bioenergetic use of these things would be a home run. As far
get
as
some students on it.
That's these are grants that I'm kind of working on and writing, but I'm not sure the funding agencies are ready to fully. They want the magic pill thing, but
so to bring you back to the magic pill, I'm kind of interested. So let's say someone like me that my diet is mostly, I eat a lot of plants, a lot of greens, broad spectrum, carrots, I eat a lot of vegetables. So I get a ton of fiber. I eat a lot of fish, and I do eat beans. So I'm not averse to legumes or even to oats. I like fiber. Let's say I'm super interested in beta-hydroxybutyrate because I've been following a lot of the research from Eric's lab, and I want to, get some of those benefits. I want to increase my FOXO3. I want to reduce my lipid peroxidation. I want some of that signaling effect. Plus, I want beta hydroxybutyrate to be around to get into the brain, things like that. Can I potentially take a beta hydroxybutyrate, whatever delivery source, powder, pill, whatever it is, and potentially achieve some of those same benefits that you would get from, you said, nutritional ketosis or ketosis from fasting?
Yeah.
Let's say, but I don't eat refined carbs, so I'm not getting a huge insulin spike for the most part. So, do you know?
So, yeah, you want to have your cake and eat it. But you don't eat.
But I don't eat cake. No, I just eat fiber.
You want to have your carbs, and that's really what we're working on right now, and understanding in a head-to-head comparison to see if we can derive the same kind of benefits from ketone supplementation as we can with a ketogenic diet. We know the first kind of convincing study of this that we did was with CNS oxygen toxicity in our rat model. And in that case, the rats were eating a high carbohydrate standard rodent chow model. And we administered via, an oral route, a ketone supplement in the form of a ketone ester. That's probably one of the more powerful forms of exogenous ketones that we've developed. And that had the ability to prevent CNS oxygen toxicity from happening for almost over 500% delay in that time to CNS oxygen toxicity. And that could not be achieved, with fasting. It could not be achieved with anti-seizure drugs. So when it comes to enhancing and preserving brain energy metabolism in the face of a tremendous oxidative stress that breaks down brain energy metabolism, we're able to enhance that and preserve that. So we've also studied in our animal model of cancer, metastatic cancer, simply giving ketones to the animals on a high carbohydrate diet. It was almost, unexpected, the level of enhanced survival that we had with ketone supplementation. This was a model of metastatic cancer, and the primary tumor was derived from a glioblastoma, a GBM, and that tumor cell line is so aggressive that when it's injected or implanted into the flank of the animal, there's rapid systemic metastasis to all the organs and the brain. And it's a model that was developed by Professor Tom Seifried at Boston College.
Didn't he publish a paper on...
It made the cover of the International Journal of Cancer. When I wanted to do the study initially, I wanted to do a brain tumor model, like with our diet and our stuff. And he sort of wouldn't let me use the model. He's like, well, use this model of metastatic cancer because it's the most aggressive thing out there, and no one will... He's like, if you create a cure, something that can cure this animal of metastatic cancer, you've basically stumbled upon something that has the potential to cure cancer. So that kind of intrigued me. So I knew working with this model would be kind of difficult because the tumor burden, the animals died so quick from the tumor burden. But it allowed us to screen a variety of things, and just using this model, understanding that in aggressive metastatic cancer, the cells are highly glycolytic, and they're highly in favor of using glucose and glutamine probably as an energy source. We have not, or looking at ways to target glutamine, but have not really kind of implemented that yet in a combination therapy. But regardless, the animals are eating a high carbohydrate diet with ketone supplementation, and it reduced tumor growth and proliferation. We think that the ketones may be altering glucose metabolism. Well, we think so. There's more sort of apoptosis in the tumors that were there, but just overall, there's just less tumor growth and less tumor burden, and enhanced, most importantly, a 50 to 60% increase in survival time in animals that are supplemented with this. And it wasn't, although the animals tend to eat a little bit less if they're in ketosis naturally, which makes a ketone supplement kind of an attractive thing for people that are dieting, because when you're sending energy to the brain in the form of ketones, it has sort of a satiating effect on your body, and it kind of shuts off that hypoglycemic trigger that makes you want to binge or crave food.
So is it changing ghrelin and leptin signaling?
We think so. Yeah, we think so. Or that's the next thing to look at. It does impact the satiety centers of the brain. It's not like you don't want to eat, but you have control over your appetite.
You know what? I just thought of lauric acid, which is C12, suppresses ghrelin in the gut, which is the hunger hormone.
Yeah.
So you might want to look into that.
Yeah.
That's kind of interesting, right?
Is that specific for lauric acid?
It's
I knew
specific.
it's certain fats. Really?
Specific
Lauric
for
acid?
lauric. Yeah.
That's interesting.
It is.
I'll look that up.
Sorry to interrupt, but anyways.
No,
Okay, so
that's interesting.
let's continue because this is really cool. So
Yeah.
So you fed them a normal carbohydrate diet, gave them this ketone ester.
Ketone ester, the same ketone ester, which is...
The tumor burden was decreased, survival time
survival time increased. Yep. And so that was, another demonstration, and the data looked very similar to the ketogenic diet. So we did this study.
They were eating less, you said, right?
They were eating slightly less, so we went back. We were thinking, well, maybe we're just getting a calorie restriction effect. Because if you have a mouse model or any kind of cancer model and you overfeed it, the tumors are going to go faster. And if you underfeed it, you're going to restrict some of the tumor growth. So they were eating a little bit less, but maybe only 10% drop in body weight. So we went back and we did a calorie restriction control experiment. And although there was a decrease in tumor, it was nothing like the ketone supplement. So ketones, we know undoubtedly they have anti-cancer effects. And it could be maybe through their expression of their gene expression, as a histone deacetylase inhibitor. We think that they inhibit glycolysis. We think that they influence a number of pathways associated with cancer growth.
Did you measure mitochondrial respiration?
Of the tumor and the tissue, or
just or any tissue. Like, so let's say they did giving them ketones.
ROS production. Yeah, it can sort of knock down reactive oxygen
in the tumor or in
production. In normal tissue, and now it's kind of interesting too that in the tumor tissue in previous experiments, I showed that it could knock it down significantly, but I think in the paper that we published, there was a slight decrease, in ROS production.
Yeah, that's interesting.
We don't know. Our experiments are sort of like a top-down approach. We find out what works, and then we're mechanistically going after it, and we're doing omics work, metabolomics in particular, and Western blots and assays. And so now we're going after the mechanism. And if we understand the mechanism, we can kind of work backwards and tweak the molecule or adjust the diet in ways that may enhance the therapeutic
It would be really interesting to do some of, I don't know if you can, radio label these ketone esters and see, like, are they being used as a carbon source for ATP? Are they being used for something else, right? Like
tracer studies. That's what we want to do. We're going to partner with a company that has sort of the market on doing these tracer fate associations. So they, several. I think even doing a 13 carbon glucose tracer fate association study where we give this and we give ketones. We look at the fate of glucose in the presence and absence of ketones and see how that may be influencing.
There you go. Is it going into the pentose phosphate?
Yes.
This is all stuff I really want to know, so I'm like super excited someone's going to be doing it.
But and I wanted to do this from the beginning, but I think we want to find out what works first, and then now that we've identified sort of things that work, with the diet and ketone supplementation, hyperbaric oxygen, I was telling you. We also we do a lot of metformin work. I have one excellent PhD student, and Nate Ward, he's looking at the effects of metformin and on survival, tumor growth, and doing a lot of the cell-based assays. And he's also looking at dichloroacetate, DCA. So it activates pyruvate dehydrogenase. So he's looking at each one individual and also in combination as a cancer therapy.
Okay, and I'll give you some of my ideas.
Okay.
So just because I want to test this. But with DCA, so I did a lot of work in my graduate research. I did a lot of cancer metabolism. I was in contact with Craig Thompson, Ralph DeBardinis. We can't link all those people that we're talking about earlier. And I did a lot of glucose withdrawal studies, glutamine withdrawal studies, blah, 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 all that stuff. So I've got a lot of interest in it stemming back from years and years ago.
Yeah.
And I also was very, active in apoptosis, working with some of the top guys in the field, Doug Green. So I sort of like the way I think about all of this and the intersection between them is that cancer cells are, here's why I think cancer cells are glycolytic. Okay. So, I mean, the Warburg effect, I know that you've talked about this and you published on it.
Is it a cause or a consequence? Yeah. Damage respiration. That's sort of
right. I don't think, and I think, even Otto Warburg himself published immediately after his original science paper, because he originally theorized damaged mitochondria were the cause of it, but then it's not. It's not that the mitochondria are damaged enough that they're not respiring. We don't really, even to this day, I don't think we have really shown that or disproven that thoroughly. I mean, I don't think it's a cause, but the reason I think that cancer cells become glycolytic, I don't know what causes it or how. I think the reason they do is because they don't want to use their mitochondria. And the reason they don't want to use their mitochondria is because mitochondria produce ROS. And that will drive, this is the whole basis of how you kill a cancer cell. Chemotherapeutic drugs, the way they work is because they induce a little bit of reactive oxygen species toxicity through a variety of mechanisms.
Some more than others, but
right, through a variety of mechanisms.
Augment oxidative stress,
which then pushes the cancer cell to death, because normal cells don't have boatloads of, pro-apoptotic. If you look at, any cancer cell, they have boatloads of it. I mean, just tons and tons of pro-apoptotics, but they've countered it and they're ready to die. You just need a little push. Yeah, and DCA activates mitochondria, and I think that's part of how
hyperbaric oxygen too.
Hyperbaric oxygen, and we were talking about this earlier. I think that's
naturally
also
stimulate it. Yeah.
Yeah, but so I'd like to see someone sort of test that, because I think that possibly DCA wouldn't be as potent at killing cancer cells if you gave it NAC or something that's going to sequester the reactive oxygen species.
So could you block it? Yeah, that's another interesting.
Right,
that would be a good control to do.
And studies have shown, that giving mice, supplemental vitamin E, something that's going to, potently sequester reactive oxygen species, actually allows tumors to grow faster. And this has been, even in recent years.
And acetylcysteine too.
NAC as well.
With metastatic melanoma, I think it came out. Yeah,
and then also there was one in lung cancer.
Yes,
I think it was the same group publishing it.
Yeah.
But so I think that part of the reason, because cancer cells are so smart, that I think that that not having their mitochondria active is very beneficial to them, because
less mitochondria too.
Less mitochondria.
So just a deficiency, and there's reports. So there's this debate. Are the mitochondria defective, or are there just a decreasing number of mitochondria? I think it's both. I think that mitochondria are structurally and functionally impaired, and I think there's a deficiency of them.
You mean in cancer cells?
I think in cancer cells, but I think in just normal cells, if you're drinking alcohol and bombarding the liver with oxidative stress, that you're damaging mitochondrial. You're damaging the mitochondria, and over time, you are going to induce the Warburg effect by causing progressive damage, mitochondrial oxidative phosphorylation. The cell's ability to maintain energy status through mitochondrial oxidative phosphorylation will be, its capacity will be impaired. So it will activate oncogenes, and the oncogenes that drive the glycolytic. You'll have some cells will die. The ones that survive are the ones that go on and activate the complement of genes that cause a transformation of a normal cell to a cancer cell. And that's how I sort of say that this progressive damage, you could do it with radiation, you could do it with chemotherapeutic agents, you could do it with inflammation. Chronic inflammation is damaging respiration. And it's that damaged respiration that can kill off cells, and the ones that survive that are hardy enough to activate the genetic program that allow them to survive are kicking on the oncogenes that will then go on and produce a Warburg phenotype. So that's sort of the metabolic theory of cancer in a nutshell. And it differs from what the thought leaders in the field are saying, that it's more of a default state to ensure the preservation of the cancer that exists. but not the cause. But there's still this elusive enabling factor that we still don't know. And I think the metabolic theory nicely explains, is a pretty elegant explanation as to how a normal cell, converts to a cancer cell. I know there's genes involved, definitely, but I mean, at the very most, the most we can link hereditary effect to cancer is maybe about 10%. 7% I think was the number that is being thrown around now, but about 10% of cancers are from a hereditary. But the epigenetics, I think, is something that and that's something that's evolving very.
Yeah, I mean, I think that
very fast.
DNA damage, that's been pretty well shown. Damaging DNA, both mitochondrial and nuclear DNA damage. That can lead to aberrant, cell cell.
And I think the
cells.
mitochondria are more important because the mitochondria have less of a robust DNA repair mechanism, and also the DNA of the mitochondria have more coding regions. So if you bombard cells with radiation, classically radiation biologists are taught that radiation is directly damaging nuclear DNA, and then that kicks on, causes the genomic instability that causes cancers. But I think what is being more appreciated now is that the mitochondria are selectively vulnerable because their DNA repair mechanisms are far less robust. They have much greater coding regions within their DNA, and they're the ones kind of calling the shots. They're making the energy. And if the energy status of the cell goes down, that's going to trigger the nucleus. That's going to trigger an energetic crisis in the nucleus, and the nucleus is going to kick on oncogenes to transform the cell from a normal to a cancer cell. So the stability of the nuclear genome is tightly regulated to the energetic state of the cell. And
so I have a little bit of a different way of thinking about it, mostly because I'm also doing a lot of research on this experimentally. And so I measure damaged DNA and I measure mitochondrial function after I induce radiation. So I'm sort of...
In primary cells?
In humans and blood cells, but so mitochondria, you mentioned that nuclear DNA have more repair mechanisms, and that's true, but mitochondria have very elegant and beautiful way of repairing damage through fusion, right? Mitochondrial fusion and fission. And this is a process. I mean, this is how we are able to repair damaged mitochondria because they're constantly fusing with healthy mitochondria, exchanging their DNA content, proteins, things like that, and fizzing back apart. So, of course, when those mechanisms become impaired, then you start to have more accumulation of damaged one because they can't repair.
Fission proteins, the production of the proteins that cause that are also tightly linked to oxidative stress, too.
There's lots of different ways to repair damaged mitochondria. That's something that I also did a lot of work on in graduate school. But I don't think it's clear. I don't think that the metabolic
far from clear. No.
Cancer.
Far from clear.
When you drop the ATP status in the cell, what happens is the cell dies and apoptosis gets triggered. And that's the primary. Before oncogenes are activated, the cell dies.
Unless it's if it's more gradual, then you have the activation. Most cells die. So you have 999 cells die, and then you have one that kind of activates the complement of genes that allows it to survive, gives it survival advantage. That's what you get with chemo too, or radiation. When you blast cells with a tumor with radiation, you get those few that can survive. And if you do that over and over to the tumor with courses of chemos, you're kind of making a super cancer because you're increasingly, selecting for the most aggressive, glycolytic, hardy stem cell-like tumor cell by hitting it with more chemo, just causing more DNA damage and more transformation and mutagenicity. So, do you see it like that? I'm not against standard of care, but I'm just
I mean, I wanted to believe in graduate school. I wanted nothing more to believe that mitochondrial dysfunction was the cause of cancer, but I just couldn't prove it to myself.
Yeah.
I kept trying and trying. It would have been so much easier for my graduate career would have been shorter for one, but I just couldn't find enough evidence to convince myself of it. And that doesn't mean that it doesn't it's not true. It just means that I just
and I
so
think
far
it's-
don't.
it may
I don't
be,
think that's the origin of cancer. I think
Yeah.
that metabolic dysfunction plays a very important role in causing cancer, primarily through inflammation, through all the effects of the insulin signaling and the inflammation, the reactive oxygen species, just all these things that are damaging the cell. But
Mhm.
I don't necessarily see it as the way that you sort of described it.
The
as
initiator.
them changing or activating oncogenes. I don't think that's really been shown.
I don't think anyone's studying that because, or studying it in the way that would make it clear. And I think it may vary between cancers like leukemia and lymphoma and relative to glioblastoma. I mean, we know these are they have a different metabolic and a different gene signature. Glioblastoma has, hundreds, if not thousands of genetic mutations, so hence the name glioblastoma multiforme. It has all these different cell types and everything. Whereas other types of, for leukemia, for example, Gleevec works marvelously well because it's targeting, something that's very specific. So I think I think it'll be impossible to get a clear answer to this, and I think maybe I'm a centrist, so I'm somewhere in between the genetic versus metabolic, but leaning more towards a metabolic origin for many solid tumors. But there's some cancers that just kind of throw me a curveball, like different types of lymphoma and leukemia, testicular cancer. And they're all responsive to chemo. Many of them are.
So what about for me, when I think of mitochondrial dysfunction, to me that leads more to neurological dysfunction, neurological disorders. Less of cancer, when mitochondrial mutations, genetic mitochondrial mutations and mitochondrial genes, there's much more, it has much more of an effect on causing certain types of neurological disorders rather than cancer, right? There's one, I think the sexually dehydrogenase 2, one of the components. I know this because I was trying to figure this out in graduate school. It was a huge
and they're really bad diseases, these mitochondrial.
But so that's what I usually think of when I think of mitochondrial dysfunction. I always think of it as being more of playing a causal or initiating role in neurological disorders and neurological dysfunction, and not so much as it plays a role. I think that mitochondrial dysfunction and abnormal metabolism plays a role in cancer, but I don't think it's that initiator the way you were describing it. I just don't think that's been shown. But
I'm going to prove you wrong.
Right.
No. Well, our lab is, we're just trying to find, the answer. And I think that, as we move forward and develop the tools, I think the answer will start to get a little bit more clear, at least using the models that we're using. But I think regardless even of the origin is important because the way we treat and prevent cancer is going to be different if we know the origin, for sure. If it is a mitochondrial versus a genetic origin or whatever origin. And I mean, there's a case for viral origins of cancer too, but these viruses are sort of the ones that damage mitochondria too. I've been sort of interested in the viral origins of cancer. It will influence, how we prevent cancer. So in addition to developing therapies, we want to study animal models and maybe inducers of carcinogenesis and then adapt them or treat them prior to or put them on a therapy prior to letting them live out their lives if they're prone to spontaneously forming tumors, or letting them live a few months prior to the introduction of a carcinogenic agent, and then seeing after X amount of time whether the tumors form. So can we prevent, this has had profound, implications for people who went through chemotherapy or had cancer in the past, and should they be on some kind of preventative there? Should they do a therapeutic fast? I get this question a lot, for 4 or 5 days to a week, every 2 or 3 times a year. Should they do that? Would it help them purge their body of precancerous cells and put that metabolic stress? And these are important questions that no one's really trying to answer, at least from a point of a prevention sort of idea. So I think that's sort of on my horizon as the next big thing. Can we develop and implement, have these protocols available for someone to do? Could be intermittent fasting. I personally like ketogenic intermittent fasting, where you're taking in ketogenic fats and ketone supplements throughout the day and through 20 hours of the day, and you have a 4-hour window where you eat a well-balanced ketogenic meal, that's rich in vegetables and, high in fats, protein. And I think that could be something that could be implemented, and something that I personally am interested in writing up a protocol for that. The studies done with metformin and showing that people who, type 2 diabetics that are taking metformin have a 62% less chance of getting pancreatic cancer. We need to study that.
Absolutely.
Should that be part of a comprehensive preventative therapy that people should do? I mean, I question, should I have my parents on this? Like, should they,
on metformin?
Or on metformin, or my mom actually had, cancer years ago. I mean, should she be on something like this? Should everybody be after, the age of 50 if most of their family members have died of cancer?
Have there been any long-term studies on the effects of metformin? Because I'm very interested in it, but I'm always hesitant with any drug or anything that's perturbing biological systems.
Well, there's hundreds of thousands, if not millions of people on metformin. So I would say, there's a long-term data out there, and a lot of retrospective studies have been done. It's relatively safe drug. Lactic acidosis could be a problem in higher doses for some people, maybe with renal insufficiency or impaired liver function. And then another thing that creeps up could be vitamin B12 deficiency. So if our ability to absorb vitamin B12 as we age is decreased, so maybe a sublingual form or even B12 injections in people that are older.
Why does metformin affect B12?
That's a question. That's a question I don't know an answer to, but I can speculate that it may influence the transporter, and it also tends to make stools loose for some people. Things go through you a little bit faster. It impairs not a good word. It changes the gut microbiome favorably. So Nature there was a paper that came out about 2 weeks ago showing that there was a favorable shift in the gut microbiome with metformin, and that may explain its type 2 diabetic, its glucose lowering effects. That sort of hit me as like, wow, I had not really given that a whole lot of thought, but it's something I think we should be looking into. So I was like, we need to collect all the, start collecting the feces from these animals that we're doing metformin on to figure out what's going on with the gut microbiome. But I think it's influencing the absorption of B12 in some way that I don't really know.
Does metformin, is it doing anything in addition to mimicking a lot of the same signaling pathways and that caloric restriction does? Like, is there something additional that,
AMP kinase for sure.
Right.
So without a doubt, I mean, it's mimicking many of the pathways associated with calorie restriction and with fasting. To what degree it's mimicking that relative to a length or duration of fasting, I don't know. We're doing some work right now looking at AMP kinase and mTOR and downstream and upstream signaling insulin and these things, and trying to get a picture of this, at least in a rodent model, and then would like to ultimately replicate some of this stuff in humans. But what I think metformin would be best used maybe in pulsing it a few times a year. A lot of these things, metabolic interventions, tend to work best when you cycle them, I think. And I really have not been doing that, but I think it's a theory that I've been working on
Why do you think
in my
that is?
scrapbook. Because your body is similar with keto-adaptation, that your body can kind of reset to that level. Initially, fasting or the ketogenic diet is sort of a stress, and it can induce a hormetic effect in gene transcription, and then we sort of get used to that. You know, our gluconeogenesis is upregulated to that level, but I think it's good to maybe pick, you know, probably not a high carb diet, but maybe a paleo diet, you know, a low carb ketogenic diet, maybe something in between, and do some intermittent fasting on occasional days. So I think this promotes metabolic flexibility. It allows our body to adapt to different situations without being kind of overwhelmed by the stressor of it. So, I mean, I think to some extent it is hormesis. And interestingly, metformin causes mitochondrial stress and actually mitochondrial damage. Some researchers coined the term, you know, that it's stimulating reactive oxygen species production and causing mitochondrial dysfunction. Metformin is, and this is kind of well known in the field. So the general feeling is that, well, if I take metformin and I go exercise, why is it not killing my exercise capacity or my VO2 max or making me lethargic or tired? It's not doing that. Actually, I think it's enhancing.
Does it affect biogenesis?
It does. So the thought that, you know, it's kind of stimulating. There's a hormetic effect. It's damaging the mitochondria. Some people believe this, and you get a secondary effect through that way. Like it's kind of like an exercise drug. But I approached it from the perspective that metformin could lower blood glucose, at least if it was high, and it activated AMP kinase, and it may decrease circulating insulin. So I approached it as a cancer drug from that perspective. But the more conferences we go to, there's a plethora of data coming out of metformin, and a lot of people are studying it from the perspective of, you know, impaired complex 1 or complex 2 activity in the mitochondria. So they're looking at it from that perspective. I know. It's very interesting.
Especially given its effects on longevity and cancer.
Yeah. And I think our most recent data did show an increase in ROS production in our cell line. So
I wonder if that's also
support
how it's killing
some of that data.
cancer cells.
Yeah, could be.
You mentioned when you were talking about gluconeogenesis, you triggered something in my mind. I wanted to ask you. I forgot. So when you're in nutritional ketosis or, you know, fasting induced ketosis, you need to make glucose. You know, you still need glucose. Your red blood cells have no mitochondria, and your red blood cells are important, right?
Absolutely.
So you're making glucose through this process that you mentioned called gluconeogenesis. And
glucose does not bottom out. It's not like one or the other. You're pulling fuel source from.
So I wanted to ask you about, like, how has anyone looked at, you know, where this so, you know, if you are on a, you know, pretty strict, you know, ketogenic diet or whatever it is you're doing to get into ketosis, does the liver use like glycerol, lactate, like both as a primary source to make glucose? And like, is that glucose predominantly going to red blood cells, or does it go, you know, has that been looked at to see like where, you know, so red blood cells, like, are they getting enough of their glucose? Are they, you know?
I think so. I mean, you'd probably have to severely calorie restrict. In those cases, you could become anemic or impair your immune system is also too highly dependent to some extent on glucose and glutamine. So you have lactate, you have the glycerol backbone, the fats. Yes. So glycerol backbone of fatty acids, or of triglycerides for sure. Lactate, yes, and amino acids. Gluconeogenic amino acids in your diet also are a source of glucose. Gluconeogenic amino acids in your skeletal muscle. You know, your muscle is constantly breaking down, remodeling, especially in athletes. So they're all sources. The contribution of each of these gluconeogenic sources in each individual probably varies tremendously. But I would say that glucose is always going to be there, and the body ensures through very powerful homeostatic mechanisms that your glucose is going to stay, rarely go below 3, maybe 2.5 millimolar, mine will drop to 4 and stay within a pretty tight range. But what does change considerably from a glucose regulation standpoint is the insulin. Insulin bottoms out to the point where I've seen enough blood work to show that in many cases, insulin and IGF-1 is below the reference range. So insulin signaling goes down. So if insulin's down, all those insulin pathways that you see on your flow charts are all going to be suppressed, and IGF-1 obviously is going to be lower. And I think that's a really important to consideration to factor in as it relates to cancer therapeutics, cancer biology, cancer prevention even, but also from the perspective of muscle metabolism. And I think by keeping insulin signaling sort of low, you upregulate factors that make you more responsive to insulin. So I think, and ketones can kind of compensate for a deficiency in insulin. And that was a lot of the, reviews by Richard Veach talked about that. And the ketones themselves are anti-catabolic or protein sparing. So if you're in a state of ketosis, you're protecting gluconeogenic amino acids in skeletal muscle from being degraded. So you are, as a metabolic fuel, but you're also, there's evidence that you're inhibiting proteolytic enzymes and pathways that would otherwise be chewing up your muscle tissue over time.
That's super cool.
So it's anti-catabolic. Yeah, so ketones are anti-catabolic in that
So then
regard.
you're probably not using, the gluconeogenic amino acids as much.
From
as you're
skeletal muscle, not as much. So the idea is that you want to keep pumping in the fat too. If you're on a ketogenic diet, if it's not sufficient with ample amounts of fat, you're probably much more catabolic. So you want to ensure that you're using the fatty acids, go to the mitochondria, they're used as fuel, they keep their mitochondria happy, as do the ketones, and then the glycerol is kind of shuttled. And it's a very nice, kind of an elegant pathway to ensure that we have that flux of glucose for vital functions like the red blood cells and there's a number of neurotransmitters and hormones that require, a baseline level of insulin or glucose to be used.
Man, we've talked about pretty much
a lot of things.
Stuff. Yeah, it's just so many things to discuss. But I'm really thankful that
we could probably talk for 4 or 5 days nonstop before we collapse.
You see how I get? I get really excited, and I'm like, okay, wait, I got to ask you this question. And I have this idea.
Yeah.
And then you're just full of information. So it's kind of neat.
A fun field to be in, right?
Totally.
I feel like I'm so lucky to be in an area of, and be in an occupation where we have the potential to discover something new that can impact the population and get paid for it.
So, because you totally, I'm sorry to change the subject, but you're talking about glutaminolysis, and I've done a lot of research on this, and there are questions that I would love to be answered, but haven't been. So, since you're looking at this and you have resources, I'll just throw it out there. So obviously you said, and this is well known in literature, that glucose and glutamine are both, cancer cells love them.
Mm-hmm.
It's crack for cancer cells, both glucose and glutamine.
Yeah.
And I've done a lot of studies on various types of cancer cells, and these are in vitro, so this is not in an animal model.
Yeah.
Where I withdraw glucose, and the cancer cells will proliferate slower. Some of them will die. But if there's glutamine there,
what's your level of glutamine?
2 millimolar.
2 millimolar. Yeah.
So then I would start withdrawing the glutamine. And glutamine withdrawal. This is all in vitro though. Glutamine withdrawal would kill them within 24 hours. But
yes.
So glutamine,
pretty lethal. Yeah.
Very lethal. And has been shown by at least some of the studies that were initially done by Ralph DeBartini's when he was with Greg Thompson, and later when he was established his own lab. where he radio-labeled and showed that actually it was being used predominantly for macromolecular synthesis and not for, which is, of course, that makes sense because a lot of tumor cells aren't using their mitochondria.
Making fatty acids, actually.
Making fatty acids, proteins, for new synthesis.
The cell membranes and stuff. Yeah.
Right. So the question for you, the question that I have, and this is, so that's one end of the spectrum. Okay, glutamine seems bad when you're looking from an in vitro perspective. And I did these studies, but many people have published on this. You're familiar with the literature. But then there's the other perspective where glutamine is really, really like your gut cells. It's very, very healing and therapeutic for gut. And when you take glutamine orally, the gut takes it. It's not getting into your bloodstream. It's not being, so...
The gut and the liver take its share, and very little of it actually gets into the bloodstream.
Right. So that's what I'm getting at.
Yeah.
The question is, if you have a mouse model of, a solid tumor that's not gut oriented, so it's not colon cancer, or, let's say it's, you've got a pancreatic cancer or
brain tumor.
Yeah. Then you give the mouse glutamine. Is that really, so is it really going to affect the tumor, or is it just going to help the gut? I mean, of course, it could indirectly affect it. But the question for me, in my mind, is, well, yeah, if you had a tumor in the gut, man, that's like crack for the tumor. Do not take glutamine. Do not, But on the other hand, if you've got gut issues,
it can be helpful.
Right? Do you see what I'm getting at?
Yeah, this is something that I've thought about. I'd stay up at night, thinking about stuff like this. Yeah,
I'm not alone.
Yeah,
because it's like, well, in vitro, it's very different because the way our bodies are working and the way glutamine, when we take glutamine, it's affecting our gut. It's very important.
Yeah,
it helps.
Yeah.
It's helped me. It's helped me with gut. But then this whole, there's this whole like conflict in my head where I'm like, cancer cells love it.
Yeah.
But the question is, if I'm taking it orally, and I have some, cancer cells in my, I don't know, my liver or something, then I guess you said liver is one thing that does use it. But so the question is that harming me or is it helping me?
Should you take it or not? Yeah, I get the question a lot. For GI cancers and liver cancer, I would say do not supplement glutamine.
Yeah.
And I would say under most conditions, I would say in those states, actually try to look up the glutamine content of food, and you might want to avoid it or minimize high glutamine containing foods. Otherwise, I wouldn't really pay too much attention. Some patients really stress out about it, but I think if you just keep your protein low to moderate, or keep your protein at a level to ensure proper, regeneration and just kind of replenishment of your normal cells and prevent protein deficiency. And being in a state of ketosis will help with that to some extent. But glutamine's pretty low on
the classical ketosis, right? Where it's 10% protein.
Yeah. And I think that will lower your blood glutamine levels. Just being on a ketogenic diet will do that. And then you could further lower it by selecting protein food sources that are lower on the end of, our glutamine, or avoiding protein types of supplements, avoiding glutamine supplementation altogether. And you may be able to further suppress glutamine by taking a supplement that's like high branch chain amino acids, high essential amino acids. So taking a supplement that is formulated in a way that kind of gives you essential amino acids, excluding, glutamine. Of course, glutamine's not essential amino acid, it's conditionally essential. But then I don't think you'll run the risk of being deficient in glutamine in any way. But I would pay attention to it if you have a GI cancer or liver cancer. And then
liver I didn't know, but GI I was definitely.
So if you have, say for example, like a brain tumor and you're taking a drug that can impair, systemically you're taking something that impairs your GI function, then it may be helpful to take a little bit of glutamine, because I really don't think the gut's going to be very greedy when it comes to glutamine.
It's very greedy.
So I think just, maybe even 5-10 grams of glutamine to help repair your gut. We know that if your gut permeability is impaired, that can wreak havoc in your body as far as systemic inflammation. So try to, there's other ways to repair your gut too, but I think glutamine may be a factor, in helping to ensure. proper
Yeah,
gut.
there's definitely other ways. I mean, I think that, like I was saying, fiber, good diet,
Yeah,
things like that.
exactly. But glutamine has been used in oncology. So yeah, glutamine for helping people with chemo, combating the issues with chemo. Yeah, I mean, glutamine has almost been like a staple,
So they give it to chemo patients that have
help them recover. It's part of the immune system too,
because your gut regulates the immune system as well.
Your gut is like 70-80% of your immune system,
it is,
right?
Yeah.
So it's huge. So we want to keep your gut as healthy as possible. And there are many drugs out there that really impair gut mobility or
diets too.
Diets. Yep.
So cool.
Yeah.
All right, Dom.
Endless amount of things.
I know, right?
Yeah. So,
just keep going.
Yeah, I've had like 100 things pop into my head about like, should I bring that in? Yeah, should I not? No.
Bring it up. I'm always interested in new things. So,
but we do have ketone esters here, and not too many people are brave enough to try them. But we have a lot of studies going on using a wide range. These are all different types of ketone supplementation, and I know that's kind of was like one of your main interests that you wanted to hit on.
Yes.
And I would say stay tuned because we have so much going on right now with like all these studies looking at ketone supplementation. And the answer that I want to really focus on is what kind of benefits are we deriving? There's been so much work on the diet, what kind of benefits can we mimic with just purely a ketone supplementation? And can we further augment the therapeutic efficacy of the ketogenic diet with supplementation? So a diet, and then we supplement, 10, maybe 20% of the calories with some form of ketone supplementation. Hopefully we work to formulate it in a way that it makes it pleasurable to taste and not taste like gasoline. And if we can do that. Does it really taste
like gasoline?
Yes. Yeah, like jet fuel, really. Yeah, it's pretty bad. But the ketone esters do. But we're working on the ketone salts that you might know of.
Yeah.
Ketocana, ketone. There's a couple ketone products out there.
Or drawbacks to taking one or the other?
The salts are like, as far as ketogenic potential, are pretty similar to MCTs. They're another level up from MCTs, I would say, right now. But they're being formulated. We're formulating them and testing them in ways to make them closer in potency to the ketone esters.
Okay.
And I think...
Are these available, to consumers?
Not for human use yet, but we're working on doing all the safety studies and then all the palatability work and formulating them in a way that probably within the next 6 months to a year that they will be out.
Oh, so
soon. We're tracking them for therapeutic purposes, so for clinical trials for specific diseases, and then to kind of work backwards to the consumer broad market eventually. But and for oxygen toxicity, obviously. That was the original application for oxygen toxicity.
I'm also really interested in it just for movement disorders. My mother has orthostatic tremor, and she is not the kind of person that is very compliant. I mean, she may try something for like a couple of days, maybe a week, That's
typical.
Very typical. And it drives me crazy because I feel like I have so much knowledge that I could help her.
Yeah.
And it's just very hard to get her to get any movement. But I'm very interested in the potential use of ketone esters or whatever delivery method, that's the best in potentially helping reduce her tremor, because, when she stands still, her legs shake, and it's very, it's inhibiting to her life. I mean, having to, when she's walking, she's fine.
Yeah.
But if she stands in line.
From sitting to standing and just.
No, even walking to standing. Just walking, standing. So standing still. It's called orthostatic tremor.
Yeah.
And it's not, I mean, it's common enough.
Yeah, I've heard about it. Yeah.
But it's not it's not as common as essential tremor.
Yeah.
But she also has essential tremor as well.
Yeah.
It's kind of interesting. But so I'm extremely interested in the potential benefits of nutritional ketosis, yes, but like I said, she's not very compliant. So I'm sort of like, okay, possibly, giving her some sort of ketone ester and seeing how that would affect. Because then again,
you might want to start with MCTs. I mean, something, what I showed you today, the MCT powder by Quest Nutrition is really pretty close, It's very potent from a ketogenic perspective. And if you were to take, 4 to 6 to up to 8 scoops a day, which would be tolerable in a course of a day, she would be in a mild state of ketosis and would be getting the benefits.
Really? Do you think that would be easier than like the MCT oils?
From perspective of GI tolerance, yeah. Many people, I would say, up to 40 or 50% of people are going to have some tolerability issues with liquid MCTs. At least a big dose that gets you up into sustainable ketosis. You can incorporate MCTs in your food, even salad dressings, cook with it, mix it in with different things. But the MCT powder I found is you can get levels about twice as higher than you can with the oil, just simply because your GI tolerance is much better in a powder form. So it's formulated in a way that kind of allows a sustained, slower release of the MCT instead of a liquid, which tends to... Some people just can't tolerate the liquid at all. I can tolerate fairly high amounts, relatively speaking, but I could tolerate much higher amounts with an MCT powder.
And you can put it and you can mix it with water, coffee, tea, whatever.
I put it in coffee.
You can maybe
and you liked it, right?
Yeah.
I mean, it's like great.
It's like creamer, but
So,
she doesn't drink.
They really
coffee
nailed
makes
that
her
product.
terms worse. That's just she doesn't drink caffeine.
No stimulants. Yeah. What about decaf coffee or something like that? Because
I think
it's a good vehicle for MCTs. I mean, you could put butter and MCT. I know you don't like sweeteners, but I just put a little pinch of stevia in there.
Stevia is okay,
and it makes it a really enjoyable drink for me.
Yeah. Yeah, that's great that it's available because I'm kind of that would be something to probably try seeing if that has any effect on my tremors.
As you know, diet's key, though.
Diet is key. It really, because if you're like eating a bunch of refined crap and processed foods and just terrible diet, it's not much that ketones are going to do. Yeah. Right?
Yeah. And what I find in people that are resistant is that if you can introduce foods that replace other foods, which is good. And I think Quest Nutrition, too, is making a line of, they're not out yet, but I've tried everything from a ketogenic Oreo to a ketogenic brownie to ketogenic chocolate bars. So these are foods that when you eat them, you are in ketosis, and they taste as good or good as their high sugar counterparts that are on the market. So I see that as almost like the next frontier, like developing ketogenic food products from whole food ingredients that are, from natural sources and not synthetic sweeteners or artificial flavors and things like that, that will allow you will ensure greater compliance of nutritional ketosis. Mainly at first maybe targeting, disease populations, but undoubtedly, people from all walks of life will be wanting to use these foods, especially if they taste good. And I could tell you from kind of a beta tester point of view that it is possible to create a line of food products from crackers to chips to you name it, really. It's possible it's pretty easy to make something taste good when you're working with fat, because fat and salt kind of make things are really good on our palate, really, and they're very satiating. So we'll eat a little bit, and it's just enough to sustain us and give us the energy that we need without overeating.
Yeah.
So I'm excited about, the ketone supplements, obviously, but I'm excited about a line of ketogenic diet food products that can ensure compliance in people who really need it. Because I see that's where there are people who know, the diet would help them from a therapeutic standpoint, but they just lose interest after trying to follow through with the diet.
Yeah, and it would help eliminate a lot of suffering, especially for some of these people with these, like you said, drug-resistant seizures.
Yeah, for sure.
Well, super cool, Dom. Thanks for speaking with me and for doing all this really cool research. I'm going to keep following your
Thanks for visiting.
If people want to learn more about your research, what you do, things you talk about, anything related related to your research? Where can they hear more about you?
I would say I'm working on a more interactive, a broader site, but for right now, ketonutrition.org I think would be the site to go to, where I compile a bunch of links on there with dietitians, ketogenic savvy, registered dietitians that I recommend, books, talks from IHMC, which I'm excited to listen to your IHMC talk this week. That's on there. So I would say that ketonutrition.org would be the site.
Ketonutrition.org. And
Yeah.
what about social media? Do you have any?
Facebook. You can find me on Facebook, on Twitter, LinkedIn. Yeah, Pinterest, maybe I go there sometimes, but Facebook, Twitter, and LinkedIn are sites where I will post information about our research or related research in the area of nutritional ketosis and metabolism.
Awesome. Thanks a lot, Tom.
Thank you. Thanks for having me.